Hi, welcome back to another Pharmacy Republic video. We're back with mitochondria again. Today we're going to look at how a commonly prescribed drug to lower cholesterol, the statins, how they can actually impact our mitochondria. Before we do that, we'll have a quick recap on how energy is produced in the body. In Douglas Adams' book, The Hitchhiker's Guide to the Galaxy, the supercomputer Deep Thought came up with the solution to the meaning of life. It was 42. I would beg to differ. I would say the meaning of life is encapsulated in this molecule, adenosine triphosphate. Everything we do is geared to creating ATP. ATP is the molecule that sustains us, that powers all our chemical, biological, electrical reactions. It allows me to stand here and deliver this video to you. While I'm doing so, I'm creating and I'm using ATP. So let's look at it in more detail. ATP is composed of three elements. We've got this adenine or purine element, which is attached to a D-ribose 5-carbon sugar ring. I've done it in red because, spoiler alert, this is an incredibly useful supplement. We will make a video to explain how it works and the uses of D-ribose, but that's for later. And that is attached to 3 phosphate groups. Okay. Now the magic happens when these high energy bonds are broken. In particular, this third one. Contains the most energy. So when this is cleaved, we produce ADP, adenosine diphosphate, plus inorganic phosphate and energy. Okay. So and we get this constant recycling. So when ATP, this terminal phosphate is cleaved, it produces ADP, which then combines with phosphate to produce ATP, and we get a recycling in an ideal world. But that doesn't always happen, and we'll make a video to explain the consequences for why, when this recycling is not 100%, it affects our energy levels. So we're back again with our original poster. So this just shows an enlarged illustration of what goes on across the inner mitochondrial membrane. So the inner mitochondrial membrane is where all the magic happens, where ATP is created. Now, the mitochondria are swimming in the cytosol of the cell, okay? And you have many thousands of mitochondria. In different organs, you have different numbers of mitochondria, depending on their energy usage. So in the brain, in the liver, and the eyes, you have tens of thousands of mitochondria. Okay, so imagine we have one mitochondria swimming in the cytosol, okay, or cytoplasm. So when we take in food, it gets broken down to glucose, and then it's transported into the cytosol. Fatty acids are also transported into the cytosol or cytoplasm. Those are our fuel sources used to produce ATP. Now, how it works quickly, I'll just do a brief overview. We've done other videos, so if you want to recap, just have a look at the other videos. So the glucose and fatty acids are shuttled across these membranes to join the citric acid cycle. Okay. Now the citric acid cycle, all it does basically is strip electrons from these compounds, and eventually these high-powered, high-energy electrons join the electron transport chain, which does exactly what it says on the tin. It transports electron in a chain-like manner. Okay. So electrons join from the citric acid cycle to protein complex 1 and protein complex 2. They are then transported to protein complex 3 via coenzyme Q10. Okay, now this is the essential nutrient that is depleted when we take statins. We'll go into further details in a bit, but for now, just appreciate that coenzyme Q10 is transporting our electrons in this chain. If it wasn't there, the electrons will not be able to complete its journey across the train, and it will form free radicals and destroy our cells. But that's for another video. So from protein complex 3 via cytochrome C, it goes to protein complex 4, where it combines with oxygen to produce carbon dioxide and water. All the while, electrons are being transported across this chain. They're pushing hydrogen ions across this membrane. So we get a buildup of hydrogen ions in this intermembrane space. When the concentration reaches a certain level, they're able to flow down their electrochemical gradient down through the ATP synthase where they produce ATP. Literally, we have a microscopic, incredibly small turbine that turns as ADP and inorganic phosphate are combined to produce ATP. Okay, it's similar to a hydroelectric dam. Water in a reservoir is allowed to flow down a cliff or, from a height where it flows through a turbine, turns the turbine and creates energy. Literally, that is the same thing that is happening in every single one of our cells in each of these mitochondria. Okay, so we learned about coenzyme Q10. We saw how valuable it is as a nutrient. Another nutrient that is incredibly important is L-Carnitine. Remember, we said that glucose and fatty acids need to be transported across this intermembrane space. So to the citric acid cycle, the fatty acids are not permeable. They can't get through these membranes. L-Carnitine is responsible for ferrying the fatty acids into the citric acid cycle. And when we use fatty acids as a source of energy, it's called beta oxidation. We get a massive 129 molecules of ATP as opposed to 38 molecules of ATP when we burn glucose. So when we burn fatty acids, we get more bang for our buck. And the nutrient that helps us shuttle the fatty acids across this membrane space is L-Carnitine. And again, we'll do another video on L-Carnitine sometime in the future. Okay, so now we know roughly how we create energy and ATP within the electron transport chain via the process of oxidative phosphorylation. Before we look at the exact mechanisms by which the statins interfere with the production of coenzyme Q10 in our body, let's look at the reasons for why they're prescribed. So as is commonly known, statins are prescribed to reduce our cholesterol levels, but that's not quite the whole story, because there's different types of cholesterol. So if you see, let's refer to another film, The Good, the Bad, the Ugly. So the good cholesterol, HDL, which grabs cholesterol from our blood and takes it back to the kidneys to be excreted. LDL, the bad cholesterol, which makes these deposits of cholesterol on the inner lining of the artery, known as the endothelium. Okay, and the ugly is the consequence of the buildup of this LDL on the inner surface to create the ugly atherosclerotic plaque, which is made up of fatty substances, cholesterol, cellular waste, calcium and fibrin. Okay, now as the LDL is building up on the inner surface of the blood vessels on the endothelium, it gets attacked by our immune system, our white blood cells or macrophages. And so they attack and they send messages to get help via cytokines. Then more white cells come. We have an inflammatory process going on. Eventually, this whole atherosclerotic plaque can develop into a clot, which can be dislodged and then cause heart attack or strokes. So the rationale for using statins was to stop or lower our LDL levels by decreasing their synthesis. We'll now look at how that is done. So what are statins? So we know that statins lower cholesterol. You may be familiar with some statin drugs. Maybe you take atorvastatin, simvastatin, pravastatin, or you might be familiar with the branded equivalents Lipitor or Zocor. Okay, now statins work on the biosynthetic pathway of cholesterol, also called the mevalonate pathway, and it inhibits an enzyme in this pathway called beta-hydroxy beta-methylglutaryl coenzyme A reductase. We're just going to say HMG-CoA reductase in future. So what happens when we synthesize cholesterol in the body? We start off with our substrate, acetylcoenzyme A, and after a series of enzymes that act on subsequent substrates, we end up with cholesterol. So statins work by stopping this enzyme here. It inhibits this HMG coenzyme A reductase competitively and reversibly. We want to have a reversible inhibition of this enzyme, because don't forget, cholesterol is incredibly important. So the body produces cholesterol, and cholesterol is the starting point for production of our hormones, for testosterone, estrogen, corticosteroids. It also is also important for the production of bile acids and vitamin D. So we do need a certain amount of cholesterol in our bodies. We just don't need a lot of LDL, the bad cholesterol. And so the statins act at this level in the mevalonate pathway, inhibiting the action of the HMG coenzyme A reductase. So before we have a look at how statins interfere with mitochondrial activity in our cells, let's have a quick detour and look at how statins were first discovered. So they were discovered by a researcher called Akira Endo and the team at Sanyo Laboratories in 1976. This team was inspired by the story of Alexander Fleming, who isolated penicillin from the fungi genus Penicillium notatum. So this inspired the team to test over 6,000 microbial strains over a period of 2 years, and they finally struck gold when they tested a cousin of Penicillium notatum, namely Penicillium citrium. So they then went on to test if this molecule which they discovered, which they called simvastatin, would reduce cholesterol in rats. Unfortunately, the rats were not a very good model for statins, because when they actually administered the simvastatins, the rats compensated by their liver producing more of the HMG coenzyme reductase. So they then went on to study the action of simvastatin in chickens who produced eggs which had an abnormally large amount of cholesterol. And when the chickens were administered with the simvastatin, it reduced the content of the cholesterol in the eggs by 50%. They then went on to test the simvastatin in monkeys and dogs, and they had successful responses. And then further on down the line, we had the introduction of statins. Little did they know at the time that they were dealing with a blockbuster drug. Now, the definition of a blockbuster drug is any drug that has a turnover of over $1 billion in a year. In fact, in 2004, the statins would generate a whopping $24 billion. So what is actually quite interesting to note is the fact that we get a lot of drugs from the products produced by fungi, okay, and bacteria. So if we look at an environment where the fungi and bacteria live together, for example, the soil, they're both competing for the same resources. So the fungi will produce chemical weapons to destroy the bacteria so that they can survive. Now, we utilize these chemical weapons in the form of antibiotics, okay? So penicillin was produced from the fungi Penicillium notatum, okay? Its mission was to destroy bacteria which were competing for scarce resources, okay? And it's also interesting to note that a lot of drugs interfere with our mitochondria. What are our mitochondria? Originally, there were ancient bacteria which were good at absorbing oxygen and producing energy. And so they became incorporated into our cell, and we have this kind of symbiotic relationship ever since, where we provide them with food and they provide us with energy. So it's just an interesting thing to note, to try and identify why certain drugs interact with our mitochondria. Is it because they think they are still have the signature of a bacteria which they're targeting? Just an interesting point to make. We'll now go on to discuss how statins actually interfere with the production of coenzyme Q10, which is the whole reason we made this video in the first place. So going back to our biosynthetic pathway of cholesterol, so we know that the statins work at this level. They competitively reversibly block this enzyme, the HMG coenzyme A reductase. So any subsequent substrates and products are not going to be produced. So if we go down this pathway, past the HMG to mevalonic acid to farnesyl, then at this stage, the final cell, we get the production of coenzyme Q10. So obviously, if we're stopping production here, it's going to affect the levels of coenzyme Q10 being produced. So that's how it can affect the coenzyme Q10 levels. Now, how does this manifest as adverse effects? Even though statins are generally safe, there have been reported side effects, and these can affect the muscle, the liver, the metabolism, and neurological side effects. So these have been bracketed under a term called statin-associated symptoms. Now, predominantly the main side effects that people find when they take statins are muscle myopathies. So a muscle myopathy could be anything from pain, weakness to fatigue. And these have been given a name, statin-induced myopathy. Now, these statin-induced myopathies and statin-associated symptoms, it's been thought that they're linked to the action that statins have on the mitochondrial activity, either via a direct action by stopping elements of the electron transport chain, or an indirect method by reducing the cofactor, the production of substrates such as coenzyme Q10, which, as we have seen, play a vital role in the electron transport chain. So, with regards to the statin-induced myopathy, what is happening is we are reducing, whether by a direct or indirect method, by maybe reducing the amounts of coenzyme Q10. This means that the mitochondria are not able to use oxidative phosphorylation to produce energy because this substrate is missing. So we can have a number of consequences. Our muscles can then switch to produce energy via glycolysis. This is where energy is being produced outside the mitochondria, and this happens when we need a quick burst of energy. For example, if we're sprinting, but as you know, you can't sprint forever because you get this buildup of lactic acid. That's the consequences of using this quick source of energy as opposed to the more efficient energy production which occurs within the mitochondria via oxidative phosphorylation. So also another consequence of not having enough coenzyme Q10 is the fact that we know it's an important cofactor that transports electrons from protein complex to protein complex. It also mops up electrons so they don't become free radicals and attack components of the cell like the membrane, which eventually destroy the cell. This is one of the reasons why people think that aging occurs. It's due to the action of these reactive oxygen species. So without sufficient coenzyme Q10 to mop up these electrons, the consequence is damage to our cells. In addition, if there's not enough oxidative phosphorylation, like we've mentioned, you get energy being produced by the glycolytic pathway. Now, the common side effect are these muscle myopathies. And the American College of Rheumatology in 2004 made a discovery that people who are getting these myopathies after taking a statin had some underlying metabolic muscle disease, such as Mercado's disease. And when they took the statin, it exposed that underlying disease. So if you are taking a statin and you are experiencing these muscle myopathies, which can be pain, weakness, or fatigue, then it might be worthwhile to check if you are suffering from any kind of metabolic muscle disease. To check for Mercado's disease, you need to check for a variant in the gene, the PYGM gene. So we've talked a lot about statin associated symptoms, the adverse effects of statins, especially on the muscle. So the purpose of this video was not to put you off from taking statins. They are still very useful medicines, and they still have a role to play, an important role to play in heart disease, because they demonstrate pleiotropic effects, which means they have actions, other actions which are not explained by its actions simply by reducing this enzyme. For example, they can stabilize the endothelium so there is no dysfunction. Remember, the endothelium is the lining of the artery which the LDL first sticks to. It can also increase the availability of nitric oxide, which is a potent vasodilator. It can prevent the immune response from developing. It also has antioxidant properties, and it can also stabilize the atherosclerotic plaque preventing it from breaking off. So they are useful, but if you are experiencing these symptoms, then it is important to contact your doctor and maybe get those tests done, okay? Another thing to bear in mind, if you are taking statins, then obviously you need to maybe consider taking some supplements, because we know from this diagram here that our levels of coenzyme Q10 are going to be reduced. So it's a no-brainer. You should be taking coenzyme Q10 if you take statins. In fact, the Canadian government has printed a warning label on boxes of statins, which actually inform people that they are at risk of lowered levels of coenzyme Q10, which can cause the development of heart failure. That is actually a warning label which the Canadian government has mandated to be put on the packets of statins. So that shows you how serious the side effects can be. But we can mitigate those side effects by simply taking coenzyme Q10. I would also recommend L-Carnitine D-ribose and magnesium, in addition to omega-3 fish oils. And of course, if you're not taking vitamin D, well, you need to do so already, especially if you're taking a statin, because we know vitamin D is actually a hormone and it is produced from cholesterol. So in a nutshell, life is dependent on the continual production of ATP in the mitochondria. The complete cessation of this ATP production is a state we know as death. Leading up to death, we can see increasing levels of mitochondrial dysfunction. And along this way, we see the manifestation of disease. For example, so if an organ is not able to produce enough energy, sufficient energy for its requirements, then that organ will start to malfunction. For example, the pancreas. If within the pancreas, the cells start having a mitochondrial dysfunction, which leads to less energy being produced, then the pancreas is unable to secrete insulin, which can result then in diabetes. Similarly, we see when the heart does not have enough energy to meet its requirements, the consequences are heart failure. So it's very important to keep our mitochondria healthy, to make sure they are always producing ATP to allow us to live a healthy life. So we've mentioned in the past the nutrients that you should be supplementing. I'll just reiterate them: coenzyme Q10, L-Carnitine D-ribose, magnesium, and also, if you're not taking it already, vitamin D and vitamin K2. We will be making more detailed videos on these nutrients in the future. To be alerted to these videos, just click on the bell icon. And if you like this video, give us a thumbs up. And if you haven't done so already, please subscribe. Thank you for watching.